No? There's only a couple more slides and then we get to the tutorial, so the more practical parts. Okay. Right. So, yes. So, one last thing on RNA-seq data, still bulk, is what distribution you should use to describe the data, what distribution better describes the data. And so you might have heard what the most common distribution perhaps used for RNA-seq data is the negative binomial distribution. So I'm going to just quickly describe why that is. So first of all, RNA-seq data is count data. I think we can all agree on this, right? You're basically counting how many times a read is mapping onto a gene. So it's not a continuous process, it's very much a discrete count. And then traditionally, the one distribution that's designed to model count data is the Poisson distribution. That describes, you have relatively large pool, relatively small chance to be picked. That's described by a Poisson distribution. However, the Poisson distribution has only one parameter, this lambda parameter, so it assumes that the variance, sigma squared there, is the same as the mean. If you look at the data, however, it clearly shows that the variance is quite a lot higher. So the more counts you have, the more the variance is also going up. And so in the plot there, the black line represents a Poisson, so it's diagonal, right? The mean and the variance are the same. But it turns out that a distribution that better represents the data, it's called a negative binomial. It's also a gamma Poisson mixture, it's the same. And so what it has is that the variance is again a function of the mean, but then it has this added parameter, this, you can call it alpha, sometimes it's called phi as well, that it's called the overdispersion. So in practice, there's more variance that you would expect by chance in just a Poisson. So that's the formula there that relates in a negative binomial the variance to the mean. And it can be shown that for relatively low variance, if alpha goes to 0, obviously that reflects again in Poisson, but that's the best distribution that describes RNA-seq data. It'll be a little bit more complicated for the single cell. So single cell has more zeros, but still, as of now, the negative binomial is still mostly used. So if you ever need to simulate RNA-seq data, you sample from a negative binomial. Right, so still kind of related to this, one thing that you need to be aware of whenever handling RNA-seq data is this mean-variance relationship, right? So both in a Poisson, but even in a negative binomial, the variance is a function of the mean. So whenever you're looking at one, you need to be aware that the other is changing accordingly. So for downstream analysis, you want to apply some sort of variance stabilization. So say you want to define highly variable genes, that's one typical step. You need to be aware that you're biasing that towards highly expressed genes as well, because typically the higher the variance, the higher the mean as well. So, or the other way around, you're doing some sort of differential expression analysis, so you're trying to look for differences in mean between two groups, for example. That also means that there'll be a difference in variance. So you maybe want to first stabilize the variance and then perform. And just an example of a variance stabilization is provided by DSeq2. So DSeq2 is one of the packages that performs differential expression. Again, it's mostly designed for bulk RNA-seq, and it provides two different functions. I'm not going to go into what exactly they do, but just to say how important this is whenever you do this type of analysis. Okay. One last thing is that you should be aware of before we get into the data is that the existence of spiking transcripts. So one way of trying to estimate the true amount of RNA in a cell is to insert these spikings. The most common ones are ERCCs. Those are synthetic transcripts that you add in the same exact concentration in each of the cells. So those are 92 single exon transcripts. They vary in length and in GC content. They vary in terms of concentration, and you add the exact same amount in each of the cells. And so essentially the ratio of just the concentration of spiking and the concentration of endogenous, so biological RNA is an indication of the initial, because you do this before reverse transcribe, I should have said. So it goes through everything. It goes through the PCR, it goes through all the things. So in theory, it's only measuring everything that's technical that's been added. And so using the ratio is a good indication of the true RNA content. Okay, I think that's...
Sorry, a question about this. Do you first always find all of those 92 again, or is it just you sample them, just a subset of those?
Sorry, say again.
If you add the spike hits, and do you get, when you measure later, do you actually find all of those, or is it often that they drop out and you only find like a subset of those?
Mm-hmm. I think you definitely find it, because that's definitely we're not gonna. I'm not gonna go into that because we're doing that tomorrow. But it's definitely the way you, for example, measure how much variance you have in the data. So you go back to where your ERCCs are following on this mean variance relationship and kind of putting back your genes onto that. But you do measure them all. They're done. Right?
You find them the way.
Yeah, you find. Oh yeah, you find them. Yeah, we'll see. They'll be in the data that will give you. So do I?
Okay, so we're gonna move to the tutorial? Yeah,
I think so.
Does everyone have internet? Well, we can start with the tutorial, right?
Yeah.
So if you open the Markdown file in RStudio.
So in the past few years, single-cell RNA-seq data, single-cell RNA-seq technologies to begin with have evolved a lot, and just in the very, very last maybe 10 years, a lot of different techniques have been developed, and this is just a figure from a very recent paper from the Sarah Tachman lab at the Sanger Institute that just illustrates all of the new technologies that have been published in the last less than 10 years. Roughly speaking, one can divide the single-cell RNA technologies into three main categories. So there is the microfluidic device, which is perhaps the newest. Well, no, not quite. There's microfluidic devices, plate-based and droplet-based. The first two are rather similar, and the idea that you get about hundreds of cells, so in different wells. So you have one cell in each of the wells, but you detect a very high number of genes. And the efficiency is the capture efficiency is rather high. You can get full-length transcripts. You can use the UMIs. We've described the UMIs, and very commonly one uses spike-ins. In the plate-based technology, the cells are first FACS sorted, so you have the extra option of sorting specific populations that you're interested in. The droplet-based, the idea is that you get thousands of cells, but you detect fewer genes. So that's the main trade-off, either many cells or many genes. The capture efficiency is a little bit more variable. It's only 3' end tag. It's very common. It's very popular to use UMIs. And there's no spike-ins. So examples of this for plate-based is, for example, SMART-seq2, you might have heard of. In the droplet-based, there's DropSeq, 10x Genomics, and others.
So are these main differences clear? So do you understand what it means?
Yeah. Okay. So single-cell RNA-seq data compared to bulk RNA-seq data is characterized by being very sparse. So there's a lot of zeros. There's a lot more noise compared to bulk RNA-seq, and that is illustrated here. So what we see here on the two axes are two different technical replicates. So they should be exactly identical. However, especially for low quantities of initial RNA-seq RNA, so the bottom there's a lot of variability. So instead of they should be exactly on the diagonal, instead there's a lot of noise.
So it means that basically, if you sequence the same thing twice, sometimes you will get a value of 100 or 1,000 counts, and the second time you will get 0, and that's just from technical noise, not actual differences in the abundance of the RNA.
And of course, whenever you observe a zero, you can't tell whether it's biological, there's just that gene's not expressed in that cell, or whether it's technical, so it was not detected, it was not captured. And this is referred to as the dropout event. That's typical of single-cell RNA-seq.
Yeah.
Okay.
So this is the main difference with bulk RNA-seq, and why a lot of the methods that have been developed for bulk data do not work. It's because the amount of noise is just so much greater.
Yeah.
And something that
more zeros. Yeah.
Yeah, so you need to think about this data in a slightly different way and just take into account all of this variation.
Okay. So let's start with the tutorial. So we'll go through just a basic workflow of the very single-cell RNA-seq data analysis. And let's first load the packages that you should have installed. So that's the first chunk there. And then that second chunk is the one that should change, because you should have your own directory rather than the one we have here.
Yeah, do you really want to just have them doing it?
I don't know. Yes?
Will the code run with all the paths that you have to your...
No, they have to change the path.
Yeah, that's...
Yeah.
Yeah, so all the images that we
You're
have,
using paste,
Yeah.
you have to remove the paste and copy your...
Yeah, that's what I...
No, you just need- you just
I did
need-
it, but I was just saying...
you just need to change what it's stored in there to your own directory.
I think, yeah, that should work. So if you set the directory at first, then
Instead of what it's there, just point that to the directory where you have
I think
saved the
that should
data.
work. It definitely does.
And then everything else should work,
Okay.
except the images. So the images
Yeah, the
won't be
images won't be
displayed
there
because
you didn't
we
download
haven't
them,
given you
so you
all
don't have
files,
those figures
but-
in your directory, but everything else should work fine.
Yeah.
And it's actually good practice to do this, right? So if you at some point decide to just change everything to a directory, you don't have to change all your code. You just need to change the directory at the first.
That working directory.
Yes.
Yeah.
Well,
Exactly. Well,
yeah.
the idea is the same, yeah. So in theory, apart from the figures, everything should run just fine.
And our Markdown usually don't like you to just set the working directory, so
Yeah.
that's why we have it in an actual variable.
Hmm.
You can use ... relevant
Yeah.
path.
Yeah, you can do that. Yes. We'll do that.
Best practice is to use absolute paths.
Okay.
Okay.
Are you all done? So can we keep going? So the data that we're going to use comes from this paper. You can open it there. There should be the link. It's a single-cell resolution map of mouse hematopoietic stem and progenitor cell differentiation. And so we just downloaded the data. So the data is freely available at the link over there, and includes row counts in the supplement. So this is all available data that you can download, and you'll have, you basically download this text file. So this chunk you don't need to run. We basically opened the text file and then saved it as an RDS file, an R specific file, and this is really just because it takes a lot less time to load in, but it's exactly the same. There's no it's not a special object or anything. It's just another way of
So
saving
the
the
code
data.
is there just for reference in
Yeah,
case you
just
wanted
in
to do
case,
it from
yeah.
the beginning.
If you wanted
When
to,
I use a scan,
yeah,
there was some simulation that takes like 3 hours. I have to save it back, all the results in the RDS because it takes too long
yeah,
Yeah.
to run it again.
yeah, it's absolutely.
But it's also compressed, so it's a good way to save your data.
Yeah.
Just
So if you
smaller
mostly
files.
using R, you really. Yes. Okay. So yeah, we saved it again as an RDS. So then you can load it using this read. So you save it using save RDS, and you load it again using read RDS. And if you look at the head of this, or the first 5 columns, 5 rows, this is what you see. So this is a count matrix, where the rows are genes, the mouse genes, and the columns are cells, samples.
You need to speak up a bit, I think. You need to speak up a bit.
Yeah. Yes.
Yeah, that shouldn't take any. Yeah, I was just reading the data.
Yes.
There's some error with the regular expression with the graph.
The read RDS.
In the line 191,
Yeah, there we go.
identical graphs, row names. Let me try to fix it then.
The line 101, you shouldn't be reading that. You shouldn't be executing that code.
No, not yet.
So the code
No,
that
no,
is
so that you have not
So-
downloaded that text file, right? So you don't have that, so you can't open
so
it.
that chunk code it
Yeah.
says do not run, and it's commented out. Do not run that.
Yeah, so the idea is that that's if you wanted, to download the proper data, that's what you would do. But then, for time, we just saved it again as an RDS file.
191.
99 to 105.
No, 191.
91. 90?
We're not there yet.
We're not done?
We are at the moment we are in loading the data, right?
Oh, all right.
In 111.
Oh, all right.
You can look at the screen.
Yeah, I mean, I'm
I
happy
mean-
to go faster. Just show that you're all.
Yeah, we can go.
Okay, so
Maybe just go through it and they can run it.
all right. So with yes.
I had an error in GC file, cannot open the connection. That's something.
So that probably means that you haven't given it the right path.
Okay.
Yeah.
Yeah, it might be the case.
Okay.
You're right. This is one.
No, actually, so,
Okay. So has everyone loaded the data in?
Can you see it? Can you open it? Can you see? Can you tell me how many genes there are in this?
Yeah, you go.
So my genes are like long non-coding or what are they?
This is just all ensemble. Genes.
Oh, it's
Yes.
a mouse, it's not
Yes,
a
that's-
human,
that's-
sorry.
Yeah,
Sorry,
yeah.
that's-
Yeah.
okay.
And how many cells? Okay.
1,940.
Roughly to that, yeah. Okay, so then we can load in the metadata. So that's the SD comma separative file that you should also have. And then, so all of these variables are categorical. Those are all information about our samples. Some of those are actually numbers, but because we want to consider them as categories, so the sequencing lane or the day that the cells were sorted, we're going to turn them into factors so that R doesn't think they're like just numbers, but we want to consider them as categories. So if you look at the head of this, for each cell, you'll have, the sequencing lane was sequenced at, the cell, the day was sorted at, the information about the mice, and the preparation day, the plate preparation day. So just quickly, this is just a quick schematic of what the experimental design for this paper was. So you should have the information about the data, but we just summarize it here. So the main questions that they wanted to answer was what changes at the molecular level between, so during the differentiation of hematopoietic sense of progenitor cells. And so what they did, they took 10 mice, all female, all 12-week-old, all B6 type, and then they sorted them in 2 different days. And then some mice were, so as groups of 4 mice were pulled together, so those are the blue and the green, the colors are just for visualization, and some 2 other mice are considered independently. And then so then you have 2 plates for each of the mice, and they were prepared in 5 different dates. And those, so 4 times 5 makes up 20 plates that were then sent to sequencing. Yes, so those are all the information we have about this samples.
So do you think that's a good experimental design? Who thinks that's a good experimental design and why? Show of hands. Who thinks that's a bad experimental design? Who knows what experimental design is? Thank you. Okay, so what questions could you answer with this data, given that experimental design?
First, I question, why did they pool them? Is it because you don't have enough data, or is it just they decided, basically? Is that something that's kind of dictated by them?
Yeah, so I would guess that it just makes the experiment easier because then you start with a lot of cells and you can just capture a lot of cells, right? But would you have any concerns about that when you're planning your experiment? So what happens if you pull all 4 mice together?
I can't correct for an effect in one of the mice.
Yeah, exactly. You lose information about
the individual, yeah.
Yeah, and so that's probably why they also perform a 1 mouse sample, to be able to just make sure that whatever they are observing when they pull 4 animals is representative of just a single animal. Yeah? Does that make sense?
It's probably also not a good idea to do plates by mouses, immediately to sacrifice them, but it would make more sense to have mix the conditions on the plates.
Yeah.
So have for each condition basically also for each day both conditions, if that's possible. I mean, they have so many mice that they
Yeah, that would be ideal, but it's probably not
Difficult.
practical in
Yeah.
terms of sorting. But that's why also you will have several plates from different animals performed in the same day.
That's what you have, yeah,
Right.
2. Yeah.
So you have 5 different days where they process these plates, and you always have at least 2 different mice in each day. So again, that's
So
a
day
balanced
It's mice
experimental
1 and
design.
2, day 2.
Why didn't they, for example, switch the single mouse on day 5 so that they actually compare 2 single mice? So now the only thing that they actually have the same day for 2 mice, that's 4 comparing against 1.
Exactly, but if you have, so one of the comparisons is to see if the pooling had any effect on your results, right? So you want to compare the pooled versus the individual. Now, if you have both individuals on day 2, you have confounded the individual with the day of sorting.
If you have all the 4
Right.
on one day, all the ones on the other, you have that confounded.
So it needs to be on different days. So remember, whenever you want to do a comparison, then it needs to be balanced across your batches. So if you're going to compare 4 versus 1, then you have to have them in both days.
The same thing with the randomized clinical trials. You control by the age, by the sex, and so on, because confounding variables.
Exactly.
Yeah. That's also, they're all female, they're all the same age, they're all the same strain. So.
So based on all of that, this is a good balanced experimental design. So you can tell that they've thought about exactly what they want to answer, what are the things that they should consider, and what are the possible sources of variation that might affect the results. So even though it's very convenient to pull all the mice, they've made sure that that is not affecting the results.
Right. There are multiple packages in R that handle single-cell RNA-seq data. You might have heard of Sera, Monocol, Skater. The one we're going to use is a package that's called single-cell experiment, and this will work with an object, a specific object that's specific for this package, that is briefly illustrated in this figure. So the main, what we already have now is the count metrics, right? So that's here is one of the assays, as they're called. The rows are genes, as we saw. The columns are samples, so cells in this case. And then you can have multiple assays. So for example, we might normalize our counts, or we might take the log, a transformation of them, and we might save that, or we might back correct our samples, and we're going to save that whole matrix in another assay here. On top of that, you can also add extra information about your genes or samples. So, for example, all those annotations that we have will get into these other matrices that are called cold data. So basically, you get extra data about the columns, about the cells, and this will be itself a matrix, right? In this case, the rows will be the samples, and the columns will be, for example, plate preparation days, sequencing lane, and such. You can also incorporate the size factor. We're going to get to that, what that is, but that will be also some values per cells. You can add perhaps externally dimensionality reduction type values. So if you perform a PCA, you can save it into the object and you'll have it. And similarly, for all the genes, you can also have information, right? You can have, where the gene is, so chromosome, or you can have the symbol instead of the ensemble gene IDs or other things. Again, you can have the spiking. So this will be basically which of these genes are endogenous counts, which are spiking, so ERCCs, as we described. And I think the good thing about this, the convenient thing, is that as you update one thing, you update everything else as well. So this avoids, so say at some point you decide you're filtering genes and you only want to keep genes that are more highly expressed in a certain threshold, you change that. You're not going to change just the counts, you're going to change also the features. So the indexes will remain consistent. So you're not going to end up with, like number one will still be the same gene both in the count metrics and in the raw data. So that is why this is convenient. If you filter out, if you index, everything remains consistent. So you don't risk that maybe you did one thing one day, then the second day you decide to change the threshold, and then your matrices will be incompatible. Yes?
One question. You reduce the dimensionality by the L2 norm?
In this case, it was just, you do PCA normally on the log,
yeah.
On the log changes, yes. So we'll get there, but yes. Normally, yes, on log scales. Okay. Right, so let's create a single cell experiment object. This is how you do this. So we're going to call this assay counts because that's what it is. So we call it SC for single cell experiment. That's how you create a new one. It says, okay, as assays for now, we only have the counts. So we have counts and we use the metrics, all counts that you should have. And then you can add already as cold data, so sample information, the metadata that we had. Yes. Then if you just say SE, type SE, that this is what you'll see. So it's an object of last single cell experiment. The dimensions are number of genes, number of cells. So yeah, this is maybe a little bit confusing. The metadata is another part of the object that's not very often used. So in this case, it's actually empty. There's assays, there's only the counts because that's all we've added. The row names will be the gene names. And you see, for example, already that the last of those genes are ERCCs by the names. The call names are the cells, the samples, yes, cells. And then in the call data, then we have the names, and those are basically the header of that other matrix. So that the one that will have cells as rows and those annotations as columns. Yeah, so cell name, sequencing date, cell sorting date, as we've seen it before. There's now no reduced dimensionality reduction yet, so we have not performed any PCA nor saved it into the object, and we have not assigned anything to spike-ins yet, so we're going to have to do that. Yeah, so again, if you now do, the way of retrieving the counts is to do counts SE, and you should retrieve exactly what we had before. Yeah. You can also, so there's currently no raw data because we have not added any information about the genes yet. So this will be an empty data frame. So data frame is just basically a matrix with annotations. And instead in cold data, we have those annotations that we added, and you see they're all factors, right? Because we made them all categorical. Yes? Okay. So we now want to add some information about these genes. So currently we only have the ensemble gene identifiers. And yeah, you always have to check that the versions are compatible. So you want to look up the paper in this case and see what annotations they have used. And sometimes it's useful to at least to use gene symbols, because that's what people are most used to rather than ensemble gene IDs. So we might want to say, instead of calling them the ensemble gene IDs, we might want to add the information about the HGNC symbol. And so one way of doing this is to use this Biomart package that you should have installed and loaded. And so if you run this chunk here, what it will do is it will create, so take the ensemble gene IDs and find what that corresponds to and add that information here.
And again, this might crash if everyone does it, because
so
it will actually...
This connects to the server, so this connects to the internet. So
and query the database, so if it doesn't work, then...
It's not all that important. Yeah. But yes, so this is nice, you don't need to have all this information.
Okay, so is this making sense so far? Is it clear what we are doing with each step?
Slower, faster?
That's great.
Yeah. Okay. That's good. Okay. So I don't know if this is working or not. Is it? Or it's just taking a while?
No, I think they're done. Okay,
so then now we have this information we can add to our raw data so that it's not empty anymore. You can add some of this gene information. And so, a way of just adding things onto the raw data is just, you call it raw data, SE, and then $, and then the name that you want to give to this annotation, gene annotation, and then you give in what we found using Biomat. So I think that should be clear. So we now have some raw data, and the raw data is the ensemble gene IDs that we started off with, but we also have the symbol, so the HGNC symbol, and the chromosome the gene is on. So if you finish, you want to do SE now. Now you should, the raw data category, for example, should not be empty anymore. So.
It's very simple.
It's perfect. It's perfect.
For 1 hour should be fine.
I mean, we ever get there. I feel much better about this one.
This goes until... We have half an hour left. Yeah. Let's keep going.
Okay. Everyone ready? No? Wouldn't appear so. Okay. Yes? Okay. So now one first thing that we want to do is we want to remove all genes that are not expressed in any cells. So basically, if one of the rows is entirely 0, so there's a 0 value for every single one of the columns, we want to remove that. So you can achieve that by doing row sums. So this will sum up of the rows, and if that's of this, the matrix, the counts as C. If this is larger than 0, we will keep it. If it was by doing that, we'll discard the ones that are equal to 0. So you can just do this. So this keep feature basically will have the indices of all of the genes that that we want to keep. So it'll be basically a logical, so it's either true or false. And so a simple way of removing, of only keeping those genes is by updating SC so that it only keeps this keep features. So those are genes, so it will be in the rows. So by doing SC of those keep feature, comma everything, we'll keep all the columns, but only the genes for which this value is true. So only the ones that have more than at least one value per gene. Yes? Is clear? Familiar with row sums stuff? Okay. Yes. Okay, so we can start with QC. So QC is short for quality control. Okay. So the first step is, assess the quality of the data we have, and potentially remove either cells or genes that do not have high enough quality. Okay, so there's numerous metrics that we can use on the cells to try and see how good they're looking. And the very first thing is library size. So basically, how many. So if we sum up all the columns, basically for each of the cells, how many reads were found in that cell. So if the cell comes from a bad quality library, will not have many reads because something would have gone wrong. A second thing is the number of genes detected. So one thing is just the number of counts, so how many counts you have, how many reads were mapped there, and the other is how many genes those reads come from. So again, this is more a measure of the complexity of the library, right? So if it's very low complexity, maybe you have a lot of counts, but they're all coming from the very highly expressed genes. That is, again, an indication of a non-healthy cell. Then some of the reads will map to mitochondrial genes, so we have not removed those. So, and too high proportion of reads that map to mitochondrial genes is also an indication of bad quality cells. It might be because the cells are undergoing apoptosis, for example. That's well, if you do have spike-ins, then that's another measure that you might want to look at. Okay, so let's try and visualize all this. So first of all, let's define the mitochondrial genes. So we can define, they're called MT genes. We can just grab out of the, so all of those mitochondrial genes will have had as chromosome assigned MT, capital MT. So you can look them up in the raw data $CHR. All the ones that will have MT should be assigned to mitochondrial genes. And yeah, if you do head of that, you'll see what they look like. Well, in this case, it's a symbol. And you can see how many there are. So if you do summary of mitochondrial genes, this will tell you how many. So all the true means they are mitochondrial genes, so the false means they're not. And we can do similarly for ERCCs. So exactly the same way, we now grab for ERCC in the row name, so in the gene IDs, and we can assign those to ERCCs. And another thing that we're going to add to the object is the spike. So we're going to tell, the object, everything that has ERCC is we're considering it as spiking. And again, we can see what those look like. And how many there are. So in this case, there's 37 mitochondrial genes and 92 spikings. Okay. So finally, so yes.
And how do you assess the value of that? Like, I mean, you have an output, a value, and then how do you interpret it? Like, as low or high, it's based on the total count?
Yeah, we'll get there. So you'll see, we'll now plot them and see the distribution and see where we want to set the threshold to keep or remove cells. So an inbuilt function of the SCE single cell experiment, or actually of SCATER, which was then integrated with the single cell experiment, and is this function that's called calculate QC metrics that will automatically calculate a bunch of metrics for you. So based on, for example, mitochondrial genes, we'll calculate how many there are. So it's just called calculate QC metrics, SCE. Then feature controls, in this case, are the mitochondrial genes. So this is what we want information on, metrics on. And %op represents the percentage. So we might want to measure, one measure could be in terms of kind of the library complexity, how many, like what percentage of our genes in a cell come from the top, in this case, 100 highly expressed genes. So you can set, in this case, I've set it to 100, you could set it to a different. So if more than, say, 90% of your counts come all from the top 100 highly expressed genes, that's an indication of a low library complexity, and so that could be a metric that we might want to filter cells on. So this function, what it does is if you assign it back to SCE, it basically calculates all these metrics and then adds them to the call data. So if after that you say, for example, the names of the call data, you'll see that it has added a whole lot of more call data. So before we had only those 5 starting point, now it has a lot more. So you'll have total features by count, total counts, total features, percentage from the top 100 features, percentage of MT, perhaps not all necessary, but it is. So some people prefer not to do this and just calculate their own annotations and only use those, but this is, so using this package does do things, makes things.
And remember, you can always check the help page for each of the functions, and then it should describe in there what all the output is in more detail.
Yeah. The most of those are quite obvious. Total counts will be, call sums, for example. And okay, so now we're going to plot some of those distributions to try and see what the data looks like in terms of those metrics that we're describing. So I don't know if you're familiar with ggplot as a way of plotting. In R, so we can maybe have a look at the very first one. So ggplot needs a data frame, so you can create it. You can make the call data into a data frame, and then you give it what you'd want on the x-axis and what you want on the y. So in this case, for example, we're looking at for the 5 different plate preparation day. The total count, the log 10, so the log of the total counts for endogenous genes, so no ERCC genes. We do the same for total features, percentage of counts from mitochondrial, and the percentage of counts from the top 100 features. So if you run this is what you should see. This is just one way of plotting this. In the first instance, we have a look at the library size. So the 5 different plots, different colors represent the 5 days of preparation. And so one reason why you might want to plot this is, for example, to see if there's consistent differences between the different plots, the different days, for example. And then in this case, we're adding a dotted line, a giant one for some reason, but at the level where that they use in the paper as the filter, right? So in the paper they say they use, what is it, 200,000? They only keep cells that have at least 200,000 counts. So in log 10, that's a little bit more than 5. And so that's where that threshold is. But you can also, in this case, we're trying to kind of reproduce what they did in the paper, but you can also just look at the distribution and decide what you want to set the threshold. And just, for example, you see this is kind of a binomial, bimodal distribution, so you might want to remove the top, the first peak on the left, because this is relatively low library size. Similarly, we can look at the rates that come from mitochondrial genes. In terms of this is very good data, so the percentage is less than 10, so we're removing virtually no cells at all. We'd be removing everything that has a relatively high percentage of mitochondrial genes, and there are none here. And then the bottom left is the total features detected. So again, they set their threshold at 4,000, I think. So again, this keeps in most cells. So this is how many genes you have at least one read in each cell. And finally, this is the library complexity, so the percentage of counts that come from the top 100 highly expressed. This is actually not a metric that I use in the paper. So that's why there's no line there. Often what people use is 90 or 95. So you want at least less than 90% of counts coming from the top 100 expressed genes. But you see that it's quite good here, it's pretty low, so that's probably why they didn't.
And so it's just important to mention here that all of these metrics are quite specific to each data set.
Yeah.
So if you have, for example, 10x data, this will look quite different. And it will depend also maybe on the specific cell types that you're looking at. So some cell types will generally have higher or lower proportions of mitochondrial genes. So there is no magic number, there is no set agreed threshold for all of this. It's more about looking at the distribution of your data and removing the outliers.
That's really why this is really important to do. You need to plot this. You don't just want to use the numbers that you've read somewhere that people have used. You really want to see this and see where the distribution is. And so, for example, you remove those outliers or maybe those outliers. So, as we said, for example, all droplet-based sequencing will have a lot more cells but a lot less genes, right? So this number will be very different. The total features will be a lot lower. The total counts a lot lower as well. So, you want to plot these things, have a look at them, and then essentially, remove outliers is the really, and it could be, and you often want to plot it in this case by plate preparation day, you can plot it by batch, by some other annotations. Okay, yeah. So in this case, we're coloring it by mouse. So those mouse
And separating each
by
sequencing
plate
lane.
actually, sequencing lane. So there'll be two plates or sequencing lane per mice. So the green and the purple, there'll only be two, because there is one individual mouse and 2 plates, and the other one there'll be 8. So it's important to see to check whether there are differences between these types of batches. So the next plot is essentially the same for, or no, we're actually plotting the ERCCs instead of mitochondria. Those for metrics, and this time we're stratifying our cells based on the sequencing lane they come from rather than the plate preparation day. And again, you're kind of using the same, checking the same numbers. We didn't put a line here.
No, it's fine. It's just to show the plotting.
Yeah.
Okay.
What's P1 and S1?
Say again?
Sorry. Yes.
It's pooled and single?
Yes. Yeah. So as you see here, we've defined this as a new variable that's called replicate. And you basically, in this case, P1 and P2 are pooled 1 and 2. So those are the red and the blue, and single 1 and 2.
That was basically for aesthetic reasons, because otherwise the legend is too wide and you cannot fit it in. Yeah.
Yeah, so this is also just to show different ways of plotting and visualizing, and ways of trying to look at the data and think about the data. Okay, so based on those thresholds, in this case, again, we're taking the paper ones. We can define a set of bad quality data, quality cells, actually, and so those are the numbers that we were mentioned. So total count endogenous, everything that's below 200,000, we're gonna discard. Total features, everything that's below 4,000, we're gonna discard. Anything that has more than 10% mitochondrial counts a mapping to mitochondrial genes, we're gonna discard. And finally, the fourth thing that they actually use in the paper is they want less than 50% of the counts coming from ERCCs.
Yeah.
So in this case, we've called this bad quality data. We can have a look at how many those are. So we see that using those 4 filters removes 244 cells, so not a lot. Yes, so we can. So we can. We haven't yet, but when you want to take only the good quality ones, you'll take all the cells that are not in this set. This was just an alternative, so we're not going to actually run this, but, oh, are we? No. An alternative approach is to use the MAD, median absolute deviation, to quantify the variability of each metric. So in this case, we're basically doing it kind of visually. We're looking at it and removing outliers. One perhaps more systematic way is to, for each of the metrics, you build the distribution, and then you look at the median. So that's the blue line there, and then you remove everything that's more than 3 standard deviations from the median.
Or you can decide the number of standard deviations, but. So that's
actually the median deviation. So you use, so the standard deviation would be minus the mean. In this case, you do minus the median because that's a little bit more robust to outliers and stuff. But the idea is that. So you take the median and then 3 or 4 median median deviation from it, and then you remove everything that's on either side.
That's what you normalize the relation between variance and mean. I mean, the over dispersion?
Yeah, so we have not done that yet. We'll look at this, yes.
But that's not important here, right? Because what we're doing these plots on is, for example, the number of genes or the number of counts. So that's completely independent.
Okay, so then we call this SC-GQ, so good quality, will be the set of SC that once we remove of those cells, so putting the exclamation mark means different from. So we keep everything that's not one of those bad quality cells. And again, we've made it smaller, so we might want to check again that we don't have any zeros. So we removed some of the columns. So. Yes. So if you type SC-GQ, so this new SC set that we're looking at, or single cell experiment object that we're looking at, you see there's a few less genes, a few less cells than there were before. We still only have counts as assays. We have now added the spiking name. So spiking names is not 0 anymore. Everything that was ERCC, we've called a spiking. And there's a lot more cold data and raw data. Okay, so it's good practice to save after performing this quality control the new SC set with a new name, good quality, for example.
So basically, if you need that to start with that data, you don't need to run the code again. You just load that.
If you're happy with this,
already.
then that's it. And from now on, that's what you work on with.
Mm-hmm.
Okay.
Any
questions
so far?
Before we move to normalization? Because it's nearly...
I think I have an older version of one of the packages. So it doesn't calculate, for example, this PCT counts ERCC.
Okay.
Yeah, so that has been changed in the newer releases.
The bioconductor gets released twice a year, and they have a lot new changes every time. So
should we
need to change? Do you want to have a break now?
Here?
before normalization since it's
Yeah.
okay. And then